Will turn.
So if I go back, maybe I can just do it here. It does, I mean, it shows here, see? But I can do this, see? I can do with the mouse, that's fine. Yeah, I can do with the mouse, and then I'll move it from here. You guys tell me.
Sorry, the cover of the presentation. Who's the volume? Assalamualaikum warahmatullahi wabarakatuh. Peace be upon you all. Shalom, Om Swastiastu, Namo Buddhaya, and greetings to everyone. Let us begin by giving our highest gratitude to God Almighty because of His blessings. We are able to gather here in good health and great spirit for today's visiting lecture. We would like to extend our highest appreciation and warmest gratitude to Dr. Sri Pujianto, Bachelor and Magister of Science, Head of Biotechnology Study Program, Faculty of Science and Mathematics, Diponegoro University. Professor Flora Tassone, PhD, today's distinguished speaker from the University of California, Davis. Professor Dr. Tri Indah Winarni, Master of Medical Science, Anatomical Pathologist, Professor at the Faculty of Medicine, Diponegoro University. Thank you for your presence, support, and valuable contribution to today's visiting lecture. Up next, we will hear welcoming remarks from Dr. Sri Pujianto, the Head of Biotechnology Study Program at the Faculty of Science and Mathematics. For Dr. Sri Pujianto, the floor is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The honorable visiting lecturer today, Prof. Flora Tasson from California University. Welcome to our campus in Semarang, Prof. I hope you nice in stay in Semarang. Thank you for your time to share your knowledge to us, and it is very useful for us to improve my knowledge and my competency, especially for my student of biotechnology study program. Jadi adik-adik semua, jadi pada kesempatan kali ini kita kedatangan Prof. Flora. Beliau adalah one of the best scientist in the world ya. Jadi kalau kita lihat jumlah publikasinya luar biasa, dengan Age Index 100 itu merupakan capaian yang luar biasa dari seorang saintis ya. Jadi ini kesempatan yang very oke ya for kita semua untuk menimba ilmu langsung dari ilmuwan yang bersangkutan ya. Beliau ahli di bidang molekular genetik ya. Kalau kita lihat publikasinya banyak sekali itu nanti meneliti kemudian melaporkan tentang kelainan-kelainan genetik ditinjau dari biologi molekuler. Thank you Prof. Iin from Medical Faculty ya. Beliau adalah kolega kita dari Fakultas Kedokteran sekaligus pengelola laboratorium kedokteran ya, Prof. Lab sentral. Jadi di sana banyak sekali peralatan yang nanti terkait dengan penelitian molekuler. Silakan nanti adik-adik biotek ini diberi kesempatan ya, Prof, diizinkan ya. Jadi kemarin sudah ada kakak-kakak Anda yang melakukan riset tentang kelainan genetik ini secara molekuler. Kemudian yang membanggakan juga saya dengar baru saja datang mesin DNA sequencer ya, Prof ya. 3,5 miliar, nanti bisa kita akses bersama ya. Oke, kita ikuti kuliah dari Prof. Flora ya, mudah-mudahan nanti kita semua bisa mengambil manfaat yang besar dari kegiatan ini. Sekali lagi terima kasih Prof. Iin, Prof. Flora ya. Assalamualaikum warahmatullahi wabarakatuh.
Thank you to Dr. Sri Pujanto for the kind and inspiring remarks. And next one, good morning to all of the respected lecturers and fellow students. Welcome to today's visiting lecture hosted by Faculty of Science and Mathematics of Diponegoro University. My name is Vina from Biotechnology class of 2023, and I'll be your master of ceremony for today's session, Molecular Technology and Diagnostics and Biomarkers, presented by Professor Flora Tassone, PhD, from the University of California, Davis. It is truly an honor for us to have Professor Flora Tassone, who will be sharing her insights on molecular technology for diagnostic and biomarkers. To guide us throughout this session, we're also joined by one of our excellent lecturers, Ms. Puspa Haning, Bachelor of Education and Master of Biotechnology, who will serve as today's moderator. To Ms. Puspa, please enter the floor.
Okay, thank you, Vina, for the introduction. Good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Okay. It is truly an honor for me to serve as the moderator for today's visiting lecture entitled Molecular Technology for Diagnostic and Biomarker. The topic is particularly relevant in today's era of precision medicine, where molecular level of innovation continue to transform disease detection and therapeutic strategies. And today we are also honored to have our distinguished guest from University of California, Davis, Professor Flora Tassone, who will soon share her expertise with us today. So Professor Flora is a professor at the Department of Biochemistry and Molecular Medicine, University California, Davis, and also a principal investigator at UC Davis Mind Institute. And she's internationally known for her groundbreaking work in neurogenetics and molecular diagnostic, particularly in the study of Fragile X syndrome and related neurodevelopmental disorders. So before we start, I'd like to share a short note that might inspire our students and lecturers. So Prof. Flora has authored around 350 scientific publications. Give applause. And has an age index of 100. A truly remarkable achievement that reflects her global scientific impact. As someone with an age index of 7 myself, so I only have age index of 7, Professor. I find that incredibly amazing and motivating, and I believe our students and lecturers do too. So we are privileged to have you today in our visiting lecture session. And ladies and gentlemen, please join me in welcoming Professor Flora Tassone with warm round of applause. And please, Professor, you can start your presentation for around 45 minutes, and then will be followed by question and answer session. So please enjoy the lecture and prepare your question for the afterward discussion. Without further ado, Professor, the floor is yours.
This one works better.
Can you hear me? Yes, now I can hear myself. Okay, thank you very much for the kind introduction. And actually, it's my honor to be here and talk to you about the research we do in my lab. We have used different molecular technology, although not all of them that I'm going to show you. So I'll give you a kind of a picture, a big picture of how the molecular approaches that you can use to diagnose several diseases. In my career, I have been working on several human disorders, but for the past 20 years, I've been focusing, as she was mentioning, in Fragile X syndrome and the associated disorders. So we have studied this disorder for many years, and I'll tell you a little bit about during the presentation. So we study with the molecular diagnostic. What exactly is it? How do we apply the molecular biology in patient care? This is the biology dogma that everybody knows. So from the DNA, you go to the RNA and then to the protein. So you have a transcription going from the DNA to the RNA, and then the RNA gets translated in protein. And you can go on the other way, where from the RNA isolated in the lab, you can, by reverse transcriptase methodology, you can make your cDNA and then do some measure of the level of a specific gene that you are interested on. So basically what you do, you do the analysis of both DNA or RNA, and combined with the different laboratory medicine in order to monitor response to therapy, but also to really diagnose diseases. And this particularly molecular diagnostic approaches has really revolutionized over the past decade what we know about the disease and how we diagnose them. So this is just to give you a general idea of the genome, just basic. I'm pretty sure that everybody knows these things, so that our genome contains nuclear genes, but also the mitochondrial genome. The mitochondria is only 16.5 kb. Is a circular double-stranded DNA, and contains mostly genes that encode for a protein that are part of the mitochondrial function. Other mitochondrial genes are also located here in the nuclear genome. The genome, the human genome, contains about 3.3 billion base pair, but as you know, the coding region is very small, account for a small percent. And in fact, you can see here that it accounts for 1 or 2% of the gene. So the remaining part is really non-coding DNA. But that contains a region that when I was a student, we used to call it junk DNA. And now we know that it's not really junk DNA anymore, because it contains a lot of regulatory element, repetitive sequences, and duplications, and also microRNA, long non-coding genes. And so it's more complicated than we really thought at the beginning. And it contains about 20-25 genes. Initially, again, we thought that we had about 100,000 genes. But then when the human genome was completely sequenced, so we realized that that was not the case, but it was also because the alternative splicing phenomena. So from the same gene, you can get different isoforms. So what it means, a different type of a transcript that will give you rise to different type of proteins. And this protein might be different for just a little bit. They might contain some domain, or they might not contain some domains. And so they might be important for the tissue specificity. So some might be in a tissue, some might be in another, some might be important during the development. So that's why the variety of gene that we have is not as as much as we thought before they were completely different genes, but the variation and the heterogeneity comes from the genes that we know because of the alternate splice phenomena. Now, just because we're going to go over some techniques and approaches, I want to just to remind you again, probably you know all this. This is the genome, and is organized, at least in the coding part, in genes. And like I may say, the genes have intron and exon. The introns are spliced out. And so they give you the mature form or the transcript, so, which is called a messenger RNA, that then get translated in proteins. And this process is very regulated, and many genes might have introns, and there are also some genes that are intronless. Instead, the ones that have introns, they have alternately spliced in different positions. For example, in the FMR1 gene, which is the Fragile X gene that is located on chromosome X, the gene span about 34 kb of the genomic DNA. It contains 17 exons, so there are 17 of this. And actually, it's interesting the fact that if you splice all the introns and you get the 17 exon, you get this transcript, which is the full transcript, which is not the major isoform in the human cells. So it doesn't account for the majority of the transcript, but less. And then you find a number of other isoforms that maybe are missing exon 12, exon 14, or they have alternative splice, like 3 different ones in exon 15. And those are the exons that contain the binding domains for other protein, because a gene encoded for a protein that is called FMRP. And this protein is an RNA binding protein that can bind to many different types of RNA, also proteins. And the way it does that is through these domains. So it depends on what tissue you're looking, it depends on what type of developmental stage you're looking at. You might have more of one isoform or more than other, depending on what the function of that particular protein is. So having said all this, what happens is, how do you get the genetic variation in your human genome? So you can have a copy number variation, what we call it CNV, and you can see that there are genes, and when they go under recombination, you can end up with a deletion because the recombination is such that you lose, for example, in this case, these genes, or you can have a duplication, and so you have these two genes that are duplicated. So they are, and this is an event of during the recombination, meiotic recombination, that can lead to this particular situation. Or you can have what we call a single nucleotide variant, SNV, or SNPs, for example, where one nucleotide changes. And so that can give you a missense mutation. So there is a change in the amino acid. And then again, it depends. If that change in the amino acid is in the really critical region, then you might have an effect. So in the sense, let's say that this change in amino acid changes a little bit the structure, the secondary structure of the protein, then you might have, as a consequence, a pathogenic event. So that variant, we call it pathogenic. Or you can have a nonsense mutation where now a substitution creates a stop codon. And what happens is the protein, instead to be the normal length, might be smaller. And so it miss probably maybe like all the 3' end of the protein, which if it contains again some important domains, binding domains, then the function of the protein is altered. But it's also, even if there is not a binding domain in that portion of the protein, now the protein has a structure that is not the one that would be, because it missed the portion. It just depends how much of the gene is missing. Or you have a repeated expansion, And this is what we found in the trinucleotide repeat disorders. And Fragile X syndrome is one of them, where we have a trinucleotide repeat element that is located in the 5' end of the gene that is a CGG. So we have a CGG repeat many times. Now, in the normal population, the mean of this size is about 30. So we have a 30 CGG repeat. But then the repeats in general are very unstable. And you find also in other disorder you might are familiar with, like Huntington disease, myotonic dystrophy, ALS. So those are all trinucleotide repeat disorder where they have this sequence that is unstable. It tends to expand with a generation. And so in the case of Fragile X syndrome, they can expand, and then depending on the number of a CGG repeat, you have molecular consequences, which means that you can have, in one case, RNA toxicity because the RNA becomes elevated, so there is more RNA produced. Or you can have lack of proteins because now the gene contains this repeat that is so large that the gene shut down. So basically it becomes silent, there is no RNA, there is no proteins, and so then you get what we call it Fragile X syndrome. So the Fragile X syndrome is caused by this expansion that goes over 200 repeat that leads to silencing of the gene, so there is no transcription, no protein produced. So the lack of protein is what causes Fragile X syndrome. And in other cases, the same, the protein is altered because there is this expansion, as in the case of, like I said, Huntington, but also there are all the spinal cerebellum ataxia, they are all trinucleotide repeat disorder. It depends on what the expansion is. Sometimes it's in the coding region, sometimes it's in the intron, sometimes in the 3' end or the 5' end. And so the consequences can be different depending also on the function and the location. So now what is these things that these genetic variant can be of different sizes. Usually the copy number variant, they are a little bit large. So we can detect them and they are more than 10 kb. But when we talk, of course, about SNVs, we're talking about one nucleotide that is substituted. But also very small, you can find also very small deletion or duplication of 3 or 4 nucleotide. And what happens is that even when you have a small deletion or duplication, you might have a frame shift. Let's say that you have a deletion of 2, now there is a shift, so the protein is completely different. And so again, this can lead to a pathological event and to diseases. And so this is basically just how you can, based on the size of your genetic variants, so what are the technology that you are using to assay them? So the single nucleotide changes, there is not any gain or loss, right? Because it's just an exchange. So the base pair is equal 0, meaning that you don't gain or you don't lose. So we're talking about just in a very small, and then the way you do it is SNP genotyping. So you can do it by a PCR, which I illustrated, or there are arrays that has a number of different SNPs that you can question and see if there is a substitution or not. And as soon as you start to go up, see, in size, you can see that you get a copy number variation until you get the trisomy or monosomy. These are the change in the entire chromosome. So you have an extra chromosome or you have one less chromosome. And then the assay that you use are array CGH, karyotyping for the chromosomal abnormality. And the sequencing, you can do quite a bit. It just depends on what type of sequencing you use. So depending on what you're looking for, then you might use different technologies. Again, if I had to think about how do we diagnose this Fragile X syndrome, we would not use any of this, but just maybe a PCR-based approach. And I will show you, probably all of you are familiar with PCR. And But initially, actually, it's interesting, before the gene was discovered, Fragile X syndrome was diagnosed by cytogenetic analysis, because if you grow the cells in a media that doesn't contain a folate, and then you do the metaphase, then you see the chromosome that is broken at the tip. And it's broken where the repeat is, but it's not 100%. So usually, or it used to be that if you see, 20, 30% of these chromosomes that were broken, then you would say, okay, there is this person has Fragile X syndrome. But then in 1991, we discovered the gene, and so then the molecular diagnostic approaches came in. And that, we have learned a lot, and that is much more precise, because now we can determine what is the size of the repeat is, what the methylation status of the gene is, and so we have a little bit more precise information. So, and this is just another diagram that is kind of the same, where you see, depending on the base pair of the mutation of the genetic variant, you can have, the different assay, going from the karyotype, the FISH, the chromosomal microarray, or CGH arrays, PCR-based method, and also the next generation sequencing. And you can see what are the things that you can assay by using different technology. So now, if we go and look at the karyotype, you can see basically if there is a structural or numerical abnormality in any of the chromosomes. So, we have 46 chromosomes, 22 pairs, plus the sexual chromosome XX in the female, XY in the male, then there are the 22 pairs. And initially, this was the major assay, the major approach to diagnostic diseases. And then again, with the advent of all the molecular technology, we have shifted, but the karyotype is still used, particularly in some diseases like sexual chromosomal abnormalities, like Turner syndrome, Klinefelter syndromes, and also to look mostly at translocation, the Philadelphia chromosomes, and so on. And you can do this karyotype G bending by using different staining that allows you to get these bands, and these bands come from the fact that the sequence is more rich in AT or more rich in CG. So there are lots of more binding on CG, then you get the lighter band, and if it's AT, you get the darker band. And so then you can really, in case of, for example, a translocation, you can see exactly, quite exactly, what is the part that has been translocated. So you have maybe part of the 14 that is on 22, and you can tell what are the genes. And then you can do also FISH, which I will illustrate in the next slides, where you can use a probe and see where the second copy of that gene is still on the original chromosome or is in the one that's been translocated. And again, this helps you to diagnose any kind of, not any kind of, but some of the disease, depending on the translocation. And in the case of trisomy or disomy, you can also, of course, use the karyotype. Now, the most common are chromosome 13, 18, but they're not that vital. They tend to die very early. And the 21 is the most common one, which is Down syndrome, and the karyotype is the one used for the diagnosis. Now, if we go instead into the fluorescence in situ hybridizations of FISH, this is basically the approach. You have a probe that has to be annealing to your chromosome, so you make the metaphase, and then some of the disease, like for example, I also studied 22q deletion. Initially, FISH was the gold standard methodology to determine the presence of a 22q deletion in one of the individual. And you can see here, for example, in this chromosome, there are 4. You have the probe that is present in both of the chromatid, and then here instead you have only on 2. So that means that the second probe is missing in this chromosome, which is the analog of the other one, the second chromosome. So that means that that portion is gone, so is deleted. And it's, it's being kind of abandoned in a way, but some laboratories still using fish for many things. So if I want to know if there is a particular protein in the nuclei of cells that carry some kind of mutation, I can also do that. And I can do in the metaphase here, on the interphase as well, where they are not divided in chromosomes. So that's why the metaphase is this and the interphase is this. And the procedure, hybridization, is just like a standard procedure where you add to the nature of the DNA that is located, the cells are on the glass slide. And then you do the counter stain with DAPI. And DAPI is this blue stain that is specific for the nuclei, so then you can see all the nuclei. And then you look at the fluorescence microscope for the dye that you're using. Sometimes you use a green or blue or green or red dye, and then you can say if there is a deletion or not. It's a target assay because you had to use a specific probe, but you can't just use anything. So if I'm looking at 22q deletion, I need to use a probe, whether it's a COMT or ProDH, one of the genes that is located in that region. So if I see the color and I see the staining, then I know that it's present. But if it's missing, then I know that it's deleted. And then by using different genes, you can determine the end point of the deletion, so how large it is. I might have an example in the next few slides. If you want to determine the presence of larger duplication or deletion, then you want to do CGH arrays. CGH arrays allows you to determine even 3 megabases, 4 megabases of DNA, they are gone. So there are lots of what we call a CNV diseases, like a 15q dupe, so a duplication of a part of the chromosome 15 that leads to autism spectrum disorders. As I mentioned, there is the 22q, and you can lose either 3 megabases or 1.5 megabases, and then and the result is the 22q deletion syndrome. And so you can think about that when you have a chunk of chromosome that is gone, it depends what's in that chunk, right? So if I have 3 megabases of DNA from chromosome 22 that is gone, there are 40 genes approximately that are located in that region. So now that region is absolutely insufficient for those 40 genes. So instead to have 2 copies, now you have 1 copy. And in some cases, 1 copy can be in a certain way buffered and can be compensated, but in other cases is too little. And so then you have a cascade of reaction that leads to those clinical features that we see in 22q deletion. And this is the same for the 16p, 16q, and all these deleted region and CNV that we know they are associated with particularly disorders. Now, it's also, if we could do CGH array, all of us, I'm sure that pretty much all of us will have a chunk of a chromosome somewhere that is missing or is duplicated. But we're still kind of walking and we're normal, I think we are. And, but it's because probably they don't involve those regions that are critical. But we don't know that. And so when we look at a certain, for example, in intellectual disability individual, one of the things you do first, in our country, in the United States, if you have an individual with intellectual disability, you have autism, for example, you immediately had to screen for Fragile X syndrome. So you rule out the presence of this, the expansion, because many individuals, they have intellectual disability, I would say from 2 to 6% have actually Fragile X syndrome. But if you have Fragile X syndrome, about 60 or 70% are in the spectrum. So autism spectrum disorder is a feature that is very common in Fragile X. So when these kids come to clinic, and one of the first things is to screen for Fragile X syndrome to rule out the presence. And then you go to the chromosomal microarrays. So you do CGH arrays, and then you determine, is that a deletion, is that a duplication in the human genome that we know maybe be associated with a particular mutation. If you don't find anything, then depending on the insurance and all those things, then you might go into the sequencing. And so say, is that sequencing that look at each single nucleotide can identify a point mutation or some kind of mutation that is already known to be associated with intellectual disability because it's a pathogenic, because it's involved the coding region of a specific gene. Most of the time, you get variation because you can imagine 3 billion of base pair. You can get a lot of different variation at the nucleotide level, and we don't know what the significance are. So they're like non-significant, SNPs, or single nucleotide variation. And then what you can do, you can see, is this variation, genomic variation, is inherited or is de novo? And the difference will be that if it's inherited, then if you test the father and the mother, you can check if it comes from there, or if they don't have it and it was just created during the transmission. There is also a possibility that there is what we call a double hit. So the mother and the father had one mutation, but it was not sufficient. And so, but then comes from the other parents, and now they are both in the child, and that double hit is what push over the threshold that you can get a disease. Or is it de novo? One of the variation is de novo. So it's complicated because you get a lot of data out of the genome sequencing, whether you do whole genome sequencing, which is even more compared to exome sequencing, which I will show you in a little bit. So, but going back to the chromosomal microarray, this is a technology that tells you what is the copy number. So do we have 2 copies? Do we have 1 copy? Do we have 3 copies? So do we have a deletion or a duplication? And it's done with a microarray. You put the DNA together, you make a library, and then you probe, and then they are all fluorescence probes that, depending on the color, they can tell you if there is a duplication or deletion or none of them. So yeah, I mean, this is just a little bit of background where you can also detect the deletion or duplication, but it's harder for the translocation or inversion, right? Because the copies are there, so you don't see it. Or it's really hard to look at the repetitive sequences. And so then you can see there is 1 copy, so this is the deletion, there is 3 copy here, and then this is of this one, and then you have duplication. And in this slide, you can see here the color that I was telling you, where you put the 2 samples together, you can isolate the RNA, make the cDNA, you label with different color. When you mix, depending on if you have a red fluorescence or green fluorescence, but you can use, it depends on the dye that you use, you can do a quantitative analysis and establish what is the relative DNA copy number in that particular samples. So the PCR-based method, you're probably very familiar with the PCR, but it's quite used in many disorders because it's very simple and it's very quick. Although there are, biased, you have 3 different steps: the denaturation, the annealing, and then the extension. So you hit your DNA first, so you separate the 2 strand, and then you anneal with specific primers, and then you extend, and from 1 copy you get 2, and then you get 4, and you get 16, and so on. So it's you can amplify a region very high yield. Sometimes depending on what is the test that you're using, the PCR is biased because it tends to amplify the smallest alleles, the smallest ones. So if you have multiple regions and then multiple alleles, and in the same sample, you might have a better yield on the smallest one. But you can optimize and then get in to see the different alleles that are present in each cells. And that depends on when you optimize your system, it depends on the amount of, for example, salt, magnesium salt that you use, or the extension time, the annealing time. The annealing time, it depends on the primer sequences. So more are CG rich, then higher the annealing temperature can it be. So if you use a higher temperature, annealing temperature, you might not get enough. But if you lower too much, then you get a specific signal. So there is a little bit of optimization, as you know. We have used it in the lab also what we call it, this is the standard PCR. There are many different variations, but this is kind of the standard that you can modify depending on your need. We have used it in the lab also droplet digital PCR, which is based on the fact that you take your DNA and you make this droplet. So in your DNA mix, you have molecular DNA that are repeated many, many times. So it just gives you a quantification without running many samples. And you run in the same tubes. And so you get many data rather than running repetitive reactions. So, and then you put it in the PCR thermocycles, and then you have a reader, and then you get this the analysis. And we use this particular technology to look at copy number, and I'll show you some example. This is the chromosome 22 that is involved in the deletion, right? So you can see there are 4 regions, A, B, C, D. This is actually the largest one, but there are also very small intercalated between these 4. And they are they're called low copy numbers regions. They are scattered throughout the human genomes. So there are many of these throughout, in many of the chromosomes, actually in all of them. And they are repetitive sequences. So there are like 10 kb of sequence that is repeated many times. So then they become what we call a hotspots of recombination. Because they are, when there is a recombination, the recombination might occur between these regions. And because they are repetitive sequences, the alignment might not be correct. And so that's how you get how you get the deletion or the duplication. So what we did, we used digital droplet PCR by using different probes. So this is the 3 megabase deletion. So people that have a quaternary Q deletion can have these 3 megabases. So from here to here, one chromosome, that part is gone. Or they can have 1.5. You can see that it's just because these 2 regions are pretty large. So the probability that there is an error during recombination is higher. And so these are the most common. So if I take a population of individual with a 22q deletion, 95% or 95% will have the 3 megabases. About 10% will have the 1.5. And then there are some few here that have interstitial deletion. See that, like this one, for example, has from here to here, or only this part is missing, but all these other probes are gone. So that means that there are other little long non-coding, basically segmental region that are repetitive, and where the event of recombination occurred, but it's less common. The most common are those other 2. And so if I do digital PCR, and I'm using 2 different probes by using, for example, this probe here, PIK4C and COMT, depending if both are there, then I know that there is not the deletion is, both are not there, I know that there is a 3 megabase deletion. But if this is here, but this is not, then, or vice versa, then you get the 1.5. And so when you do the analysis, you look at different genes here, see, if they are present or not, and then this is a copy number in the 2 copy number in a normal individual, but then you have some that have the 3 megabases deletion and some that have 1.5 megabases deletion. So you can do CGH array and do the copy number variation by SNP, so you can use this technology, or you can use FISH. Now, because FISH was the very first approach, but was much more laborious, and now the PCR base are a little bit more common, easier to approach. If you have the machines and the access to the technology, then it's faster. So this was a kind of a PCR base. Then you can do Sarin blot. Sarin blot is done for several diseases, and we have been using this for 20 years to diagnose Fragile X syndrome. And so the way it works, you have your DNA that you isolated from, you can isolate DNA from blood, from tissue, from cell culture, from whatever your source is, and then you digest your DNA, and usually you digest with some enzyme, and the enzyme that you use are specific for your disease. In our case, when we do the diagnosis of Fragile X syndrome, we use EcoR1, which is one of the most common restriction enzyme, coupled with one restriction enzyme that is methylation sensitive. And the reason is because in Fragile X, as I mentioned, if you have a full mutation, your gene becomes methylated. So the enzyme will not cut if the gene is methylated. Will cut only if it's unmethylated. So if it's methylated, it will not cut when you run on the gel. So when you basically have your digested DNA, you run it on the gel, you blot the gel, and you transfer your DNA on the membrane, and then you hybridize this membrane with a probe that is specific. So it's a piece of DNA that is contained within the FMR1 gene, so it will anneal. But if the DNA was not cut, the band that you will see is much larger. But if it's cut, it will be much lower. And so if it's cutting, means that it's unmethylated, and probably you don't have, obviously, the full mutation. But if it's methylated, you see this band up there, you say, okay, this person has Fragile X syndrome. And sometimes, I think like more than 50% of the time, some of these individuals show a pattern that is a mixture. So you have the full mutation, but also you have some alleles that are non-methylated, and we call it mosaic. Now, mosaicism is very common in human disorders, and you can see mosaicism in autism, in Fragile X syndrome, and in many other disorders. So that means that the mutation is not fully what you expect or what is causing the disease, but there is a variation, and that what is important to detect mosaicism, because the part that is actually either non-methylated or is not mutated is something that might lead you to a normal functional protein. So it's not completely absent, so the expression is not completely altered, and so the phenotype of those individuals might be a little bit better off of those they have a complete mutation, whether it's methylation, whether it's expression or other things. So I like to use in our diagnostic, as a diagnostic tool, the Sarin blot, because I can see it well. But it is a long process, so it usually takes 3 or 4 days. You have to be good in doing it, because having background is really easy. And if you have a background on the gel, then on the film, you can't really see very well, particularly if you have to assess mosaicism. And also require quite a bit of DNA. So if I run a PCR, I can use between 10 and 100 nanograms of DNA. But if I run a Sarin blot, I'm going to need 10 micrograms of DNA. So it depends, also what are your samples that you need to diagnose, that you might not have enough DNA. So it might not even apply southern blot. But if it does, you have to have enough DNA in order to run the test. So many labs are kind of abandoning because the PCR based approaches are very competitive. They don't give you all the, I would say, quantitative results, but they are still giving you the results. And so it depends how, so if you are doing diagnostic testing for a person, you say, okay, this is a person that has Fragile X syndrome, maybe has mosaic, so it shows some alleles that are methylated, and leave it as that. But if you are actually doing research and you are looking at the cells and the expression of the gene, I mean, you really want to characterize what is the molecular basis of the mutation, if there is, if there are a methylated allele, how long they are, how many cells carry the mutation, how many cells carry the unmethylated allele, and so on. So then I think the southern blot is a better approach. Now, if you have the polymorphism in human DNA sequences, we mentioned, can be the SNPs, so one substitution can have simple repeat or structural variants. And then you can do, again, a SNP analysis. Again, we have chips from Affymetrix or other company. And there are so many of SNPs. So if there are basically there is a single nucleotide variant pretty much every 300 base pair. So in the human genome, we have a lot of them. And the majority are not doing anything, so they don't have really consequences. But other can be predisposing to diseases. And a lot of study have been done with looking at the entire genome and can I see a certain pattern. So usually in the years, maybe 10 or 15 years ago, we were doing gene identification or gene association study with an hypothesis. So I hypothesized that dopamine gene and the polymorphism, depending on what kind of polymorphism in allele you have, might be associated with autism. So there was an hypothesis. Then we went to the wide genome using the SNPs analysis, and at that point there is not an hypothesis. You kind of go, I don't say fishing, but kind of. So you look at the entire human genome rather than looking at one gene and say, can I identify the variation in the nucleotide that define a pattern that is much more common in individuals that have autism or individuals that have intellectual disability compared to the control samples? And so this has been given quite a bit of good results, for example, in schizophrenia, but also in autism. There are some profiles, some genes that have been identified, and they say that SNPs is associated with a particular disorders. And so as you can see, it's just a single nucleotide variation. Oh, so this probably should I have said it before. So this is what I was mentioning before about the copy number variation. So these are what we call a segmental duplication that I call low copy repeat. So those segmental regions, they have these repeats. So during the recombination, you can have a misalignment in the crossover. So because these are the same, they contain the same repeat. And if they don't align correctly, the result is that you get a duplication over the deletion of the gene. And as I said, there are many of these segmental duplication. And again, that's why many of us probably have a duplication or deletion in our chromosomes or in our human genome. But if they don't affect region that contains many genes or particular genes, then we're okay. But in some cases, and actually in many cases, they can lead to disorder and diseases. This is, for example, was a case that we looked in autism where the kid had this deletion of quite large on chromosome 4. And then we looked at the parents, and the other brother did not have it. And the parents, so we said, is it de novo or is inherited? So this is the region that it was deleted in the child, and we found it was also deleted in the father. And but then the father didn't have autism spectrum disorders. But then when we went and we did the sequencing of the human genome of these individuals, we found a couple of variants in the mother. They were not in the father, but now they are in the kid. So it's not 100% sure, but is what we call a double hit that I was mentioning before. So now you have two different mutation that might lead to a disorder, but if they are separate, they are not sufficient, so they are not enough. And almost running out of time. I just wanted to show you a little bit the sequencing. So initially, when I was your age, I was doing Sanger sequencing, running these big plates with acrylamide gel and loaded the different 4 nucleotide, and then read here, like in this gel. It was actually kind of fun. You get these gel acrylamide that you blotted on the autoradiogram, and then you get all these lines, and you start to read A, T, C, G, and so on. Now it's much easier because there is a capillary electrophoresis that gives you the electrophotogram that reads for you. You need to check, and so you can sequence anything. You just can use the primers and do smaller region, or you can do these next generation sequencing approaches. They are like exome sequencing or whole genome sequencing, and you have to make some libraries, and then from the libraries you can determine the sequence. So you have to prepare your samples, and then the difference is that you obtain thousand and thousand of reads. So it's not just one time, but you get that same region. You get maybe, depends on how in depth you want to go. You can get 30x, 50x. More X you have, more expensive it's going to be, but more precise it's going to be. But you get, the alignment and one nucleotide 30 times, and maybe even if 1 or 2 times is different, you know that the majority is correct. So, but then you have to check, of course. And you determine, you get a lot of data there, particularly from the whole genome sequencing, because the whole genome sequence is 3 billion of base pair. The exome sequencing is only the coding region. So we're talking about 1.5 to 2%, but you are missing all the sequencing for the microRNA, the element, the regulatory element, whether they are silencer or enhancer. So you lose a little bit of that, but at least the genes are sequenced. And in particular disorder, many cancers disorders, there are panel of genes. So instead, when you do the sequencing, instead to get the sequence of the entire genes in the human genome, you sequence maybe 40 or you maybe 60. Those are the ones that have been associated, for example, with colon cancer or with testicular cancer or whatever cancer. And so then you focus on those because the majority of the mutation have been found in those genes. So then you get less data, you're more specific. Some genes have multiple sites where the mutation can be. And so these custom-made or panels are also very, very useful. And so this has just, the exome is just the exome, and here instead in the whole human genome, you get to also the region in between. That's just a diagram. And then there are different platforms. There are different type of sequencing that you can use, different chemistry, and there are the pros and cons. Cost, how much, how is the length of the sequencing that you get, what is the depth in the in the sequences that you see, so how precise can it be? So you can see that, depending on what you're doing, also there are the ones that are specific for methylation, there are the ones that are specific for alternative splicing, so they can get all the alternative splicing isoforms. Depending on what you're doing, then you can customize the type of platform that you can use. And again, here, the difference between doing genomic DNA sequencing with a Sanger or the second generation sequencing, that you get more read. They can be used to assemble larger. So, when you you do the sequencing by Sanger, then you get maybe 400, 500 base pair, but here you can get KB, and now, because I'm running out of time, I'm just going to stay. But these are just the different, different ones that you can use. But I want to mention also the nanopore. Nanopore is the most advanced type of sequencing that allows you to get larger. You can get up to 200 kb of sequencing, which is, for example, if I'm looking at repetitive sequencing, I can go through that much and see, because the repetitive sequence has always been a kind of a mystery. Is that anything inside? But you can't tell, because then when you start to do PCR, you don't know where you are, because the repetitive sequence doesn't allow you to go through. But now with the nanopore sequencing, you can do that. And so you get the much longer sequences. And so we have been going through all this, but going through different technology and in 20 years. And then, the nanopore is the one that is more, more use, that people tend to give an approach. Even in Fragile X syndrome, there are studies that are looking at the nanopore, so you can just do a sequencing and say there are 200 repeats, or there are 100 repeats. But again, it's just a matter of to optimize. Then I want to just tell you a couple of things. There are high throughput technology, so you can look at transcriptomics, proteomics, interomics, metabolomics, microbiomics, and phenomics. I mean, there are so many. And so what you do, you look at the entire spectrum of expression of RNA, or the proteins, or the metabolites. And so you interrogate, when you are comparing disease or looking at disease, is there a profile in the RNA or in the metabolites or in the proteins that is different between those that have a disease compared to controls? But you're not looking at one, you're looking at the entire profile. And you can say, okay, in these disorders, I have a lot of protein that are part of the mitochondrial pathway, or part of the calcium pathway, or whatever, coagulation pathway. They are really different. So there is a downregulation or upregulation, or a number of proteins, or a number of metabolites, or a number of RNA that I find much more frequent in those individuals that have my target disease. And so you can do those studies as well, and they're very useful. And so the molecular diagnostic, as we started, have really transformed the medicine, because you can do testing and ask and answer the question, is this baby healthy? What disease is this patient at risk for? Does these patients have a disease? What drug should I prescribe as the disease returns, so can get back? Or can I use it to say, let's say that you have a disease and you can be treated with different type of medication. Some medication might work for somebody, but might not work for somebody else. And that's why, we share my last slides, then we go into the personalized medicine. I always think, like, when I have a headache, if I take Tylenol, it's not doing anything. But if I take ibuprofen, 20 minutes and the headache is gone. So I guess my body, my background is such that I respond personally to that drug, but not to the other. Now, every single drug is metabolized by the CYP40 genes, and there are many. Some are extremely metabolizer, some are low metabolizer. So is it possible that maybe I'm an extreme metabolizer? When I take Tylenol, I chop up the drugs right there, and so doesn't have enough time to act. But now this is a simple, a very silly example, but think about who has cardiovascular disorders and they go to warfarin or all those drugs. So it's more personalized. So if I can tell you have this profile, so don't take this medication, take this other one, or you go into a clinical trial, maybe that's not the drug for you, maybe you should get to this other one. Also then don't go into the clinical trial. So that's the precision medicine that we're going towards. Then we don't, basically where every patient's benefit from the individual treatment, not some kind of, this is just a general approach where everybody gets the same things, but in some cases it works, some cases doesn't. And stop talking. Thank you for your attention.
Okay, thank you very much, Prof. Flora, for such insightful and inspiring presentation. And your talk has given us a clearer view of how molecular technologies advance diagnostic and biomarker development. And we can conclude that molecular diagnostic can be applied because each condition express different molecular pattern, whether it caused by CNV or SNV, that can lead to molecular consequences that leads to pathological condition. And we have learned also that there are many methods that can be applied to molecular diagnostic, especially the very common is PCR and then FISH microarray, and depends on the size of the base pair of the DNA, and the last is sequencing and next generation sequencing. And molecular diagnostic transforming medicine, and we still have room for exploration of biomarkers to achieve the precision medicine instead of one size fits all. Okay, so maybe we can move to the stage for the Q&A session.
And while I walk, I just wanted to kind of emphasize that you guys are doing biotechnology, and it's important, of course, to use this technology to determine what the disease is. But also what you want to do is, okay, I determine the disease, what can I do about it, right? So can I develop, when I was talking about this profile, can I determine, can I identify that biomarker that can help me to determine, for example, who is going to develop that disease, particularly all the late onset disorder that you don't know who is going to get it. And so can I predict, based on this profile, who is going to be a more risk to develop the disease, who is going to be the ones that have a more severe, how progress is going to be rapid, because sometimes right now we don't have that answer. And so that will be, particularly again, for those diseases that are late onset, like the neurodegenerative disorders, can we predict all these things before the symptoms appears? So then you can do something, even if you can't cure it, but can you slow down the process and make the quality of life of this individual much better?
Okay. Now, thank you very much. Okay, we will now open the floor for question and discussion. For those who wish to ask, please raise your hand and mention your name. Okay. 3 participants first. Hanifan. Another one? Okay. And then the last one for the first session. Okay, maybe 2 participants first. Okay, please. Hanifan and Hanifan first.
Okay, good morning. Thank you for the opportunity first. My name is Hanifan. I'm from Biotechnology 2023. Actually, I have some question about the last slide about the future of molecular diagnostic, which is in precision medicine. All right, so I want to ask about, Professor, what research you've been working on recently. Is it related to precision medicine or is it not? And what's the main challenge that, for this topic? I mean, for the precision medicine. What's the main challenge on working through this topic? Have you ever thought of estimating when we could reach this precision medicine into the optimal version of it? Thank you.
So if I understood correctly, if I'm using precision medicine, the first question, if it's something that I'm currently working on, and then what would be the challenge in using it. So let me tell you a little bit in general. So precision medicine requires that you have a very good idea of what the background, for example, the genetic background of a person is, looking at different genes, and the genes that might be related to that particular disorders. I think at the moment is not into the healthcare system, so it's at the research level. And particularly is more diffuse in cardiovascular disorders. So where you look at some of the polymorphisms that you have in some of the genes, and then you can determine what is the best medication to go for. What is an anticoagulant or things like that. I think it will take a little bit of time, because when you go to the doctor and the cardiologist, and they say, I have this problem, heart problem, they're not going to check all these polymorphisms. So those are kind of a research study, but they are pushing, because then you can say that, the advantage is the care of the patient, so that will be way better, even in terms of cost. For us, in my lab, what we're trying to do, because I study the associated condition to the Fragile X gene, and one is neurodegenerative disorders. We are looking at the, like I was mentioning, at the profile, and looking at identify some proteins and some metabolites that can tell me who is going to be the most at risk of developing that disorder. So I can act upon before. But even if you identify a metabolite, that you can say, these metabolites changed since he did not have the diagnosis of that disorder, and then he get the diagnosis, now it's changing it much higher, okay? But then how do I target to make it lower? And if I can rescue, do I really rescue also the phenotype? So you might rescue the elevation in the plasma level, but then it might not correspond clinically to benefit in the clinical phenotype. So this individual, for example, have a tremor, have ataxia, and so then you say, okay, maybe that metabolites affect the mitochondrial function. I reestablish a little bit, but still the guy is going to have tremor and ataxia. So it's not that easy, but that's what we're going to. And so that will be more personalized, because that might help one person, but not the others. So that's what the challenge is.
Okay, thank you so much for the answer, Professor.
Okay. The next participant.
Good morning, Professor. Thank you for the lecture that you have given before. And my name is William. I want to ask about how we do the diagnostic with molecule for the prenatal baby, maybe, because we know baby have genes from their parents, both parents, and they have the different genes, and how we checking on that, because the genes of the mother cannot 100% be in that baby, because that is also from the death itself. And I want to ask how that the mechanism of the prenatal molecular diagnostic for the baby. Thank you.
What exactly? I'm not sure I catch the question.
The molecular diagnostic for prenatal condition.
In general?
Yeah, general,
Yeah.
General.
So in general, the molecular diagnostic.
Yeah. How the mechanism that is working, we use the molecular diagnostic to checking for the prenatal.
Yeah, so it depends on what kind of things you're looking at. There are different approaches. Of course, you do prenatal when you do the looking at the amniotic liquid and see, oh, you can look at it now in the plasma of the mother, looking for this piece of DNA, they are circulating, and looking at the level, looking at the type of, so there are different things, but it just depends on what you're looking for. But definitely there is a lot of work that has been done because obviously we have a lot of diseases that we don't diagnose, particularly all the neurodevelopmental disorders that we can't diagnose at the prenatal level. What we do, for example, in Fragile X, we do amniocentesis, and then you can see if there is an expansion there, but that depends also on what kind of week you do it. So if you do a 13 weeks, you can see the expansion, but you don't see the methylation. So then you don't know exactly, you know that there is something that is not in the normal range, but then it can be defined. So I guess that there are many approaches that you can do, but there's quite a bit of work that still we have to do it.
Okay, thank you, Prof.
But those, the plasma level with all these molecules, exomes that are going on that you can test, they are very promising. And so they've been using, even the knuckle from the size in Down syndrome, for example, it's one of the things that has been used. People have been using.
Okay, maybe just thank you, Professor.
Already, okay. Maybe I will add the question from the participant, especially for NIPT. Usually we use NIPT to detect the potential disorders of the baby. Usually, what kind of method that we use? Is it sequencing enough, or we had to dig into deeper method that to anticipate and define the disorders? Yes. Okay. Okay, maybe next participants. Any question? Okay, Fina.
Okay, thank you for the chances that have been given. I'm Fina from Biotechnology class of 2023, and I've been learning about short tandem repeats, and from what you have presented, CGG expansion in FMR1 can reach over to 100 copies and lead to methylation and silencing. I was wondering what makes the CGG sequence itself so unstable compared to the other STRs. Thank you.
Sure, you have the right questions. So it's just that we know that there is instability, and the instability is due or is influenced that the number of CGG repeat. So more larger, they get more unstable. And also within the CGG repeat element, there are some interruptions that are called AGG. So let's say that you have an allele of 30 repeats, most likely you have a 2 AGG interruption. So that CGG stretch is not pure. There are 2 AGG. Now, when the CGG repeat element tend to increase in size, those interruptions tend to be lost. And we know that they affect the stability. So let's say we have done many studies on this, where we looked, for example, at females that have a permutation allele, so mothers. And the mothers, then the question is, oh, I have a permutation allele, what is the risk that my kid will have a full mutation? And so for many years, we always said, well, it depends on the CGG repeat number, such that if the allele is about 90 repeat, almost 100% of the time, the kids, if they will inherit that allele, will be in the full mutation range. So greater the CGG repeat number, greater is the risk. But if those alleles have AGG interruption, the risk decrease, particularly in this, in a certain range. And there is quite a bit of difference in a range between 65 and 85, where if you have a pure stretch, you might get an allele in the full mutation range in the next generation. But if you have 2 AGG interruption from, let's say, 80%, the probability dropped to 20%. Which, so if you're doing genetic counseling, this is very important for the family. and they can make, informed choices. But, they tend to be unstable. If you have a cell culture that has cells with a repeat allele, then you grow and you pass it many times, you can see that eventually it will move and it gets bigger. And sometimes it gets shorter, so there is retraction, but that's a different mechanism. And we think we have some data that shows that the instability is also related to some polymorphism in in DNA repair genes. So some of these genes, like in an Huntington, in myotonic, in ALS, they seem to affect the stability of the gene. So if you have a certain particular polymorphisms, the probability that the allele is unstable is higher than in other cases.
Maybe any question for 3 participants? Please raise your hand. Oh, if doesn't anyone?
So how many of you are using these technologies, any of them, or are familiar with, PCR, sequencing, RNA-seq? Have you started or you're kind of at the beginning?
Yeah, PCR is very common in here, so we still using PCR for daily practical.
to do it
in the lab for the practical practice. Professor, maybe I want to ask one question before we close the session. Okay. Maybe I want to ask one question before we close this session. You talked before about the cell culture model to fragile eggs model. I previously developed the in vitro model of neurodegenerative disease using HEC293 cell line. And for the neurogenetic model, we can use what kind of cell line, and we have to how many passages that we need to develop the neurogenetic models. Maybe something like that.
So you're doing using the cell model?
Yes, previously. In my previous lab, I developed the in vitro model for neurodegenerative disease, but still in common.
Right.
Yes.
Well, we use the cell model that we use is from fibroblast. So from skin biopsy, we make the fibroblast primary cell line, and then we have the lab that does stem cells to basically reprogram the fibroblast in iPSC cells, and then they can get neurons by differentiating them. It's not that easy because, even if I look at all the molecular bases that I find in blood directly compared to fibroblast, the fibroblast doesn't show as much as the toxicity that we see in blood. However, it's a good model to create neurons and other cells, and now we are going into organoids, which is even more complicated. But can create some area of the brain that you can study.
Maybe how can we evaluate maybe the molecular pattern of the models, especially the biomarkers that mimicking the disorders?
Right, but also you have to keep in mind that those cells are not in the environment where you have in the cell body, particularly if you're looking at neurons. They have astrocyte, oligodendrocytes, and so you mimic a little bit. But that's what we can do. I think it's a good way to study how the pathway and the molecular mechanism might affect these cells.
Can we use the marine, I actually study about drug discovery in marine-derived bioactive because they have a unique structure. And is it possible for us to use the bioactive to study the modulation of disease prevention, maybe?
Yeah, that would be a kind of way to go, I guess. So if you can find the way in the system to do that you can monitor how. Like for example, when you reprogram the cells in Fragile X, they are unmethylated. So they are not methylated. So then you start to grow and you start to see how they methylate. So you can study actually the process, how it occurs, that you can't do obviously in humans.
Thank you very much. Oh, Another question? Yeah, okay.
Hello, Professor. Thank you so much for your explanation. It's very insightful. My name is Arina. I'm a lecturer of biotechnology study program, and this your explanation is really intrigued me because you mentioned about autisms, and I've watched maybe you know a series called The Good Doctor. He has a yeah, he's a doctor and he has autisms and also a Savant syndrome. So my question is based on that series. The first question is for molecular diagnostic in autisms, and we know that it has many wide spectrum or different symptoms or different subtypes, and how can this variation can be detected through molecular diagnostics? And then the second question is, since if there is the comorbid situation like in this movie, they have autisms, and if I'm not mistaken, he also had a Savant syndrome. How then this neurological condition like Savant syndrome can be detected by this molecular diagnostics, and is it possible or not? Savant. Thank you.
So if you have, usually the autism spectrum disorder is diagnosed at the beginning clinic, the behavioral, because it's not like Fragile X, there is a gene. In autism, obviously, is a very genetic based, but also environmental and also gene environmental interactions. Unfortunately, I just had to make this comment. In our country right now, it's kind of a mess a little bit because our administration is pointing things that they are not, how would I say, they're not real. So I don't know if you guys have heard that our administration is putting out there that is Tylenol during pregnancy that is causing autism, which is a kind of a problem because really we know that there is a very high genetic component on autism, but it's not one thing. So if you look at the case of autism in general, maybe we can explain 40 or 50% of the cases by looking at copy number variation that cause autism, a point mutation in different genes. We know that some of the infections during pregnancy or pollution during the exposures, they can lead to autism. But we also don't know a lot of cases that we don't know what they are. But usually, as I mentioned, if you have a cohort or an individual that come to clinic with autism spectrum disorder or intellectual disability, you would do those tests. So maybe you rule out Fragile X. Okay, there is no Fragile X. Then you do copy number variation. There is no a deletion or duplication. That is known. You do sequencing. And sometimes, some people says, well, you have identified a mutation in this gene, and then what? Well, but sometimes for the parents, it's really important to have an answer. They go through the odyssey or try to figure out what's wrong with their baby, and finally they have the answer. But that's basically kind of the itinerary that you follow, and you try to find if there is a mutation. And then, but also keep in mind that all these diagnostic tools that we have to molecularly point in that direction, they had to be coupled also with the behavioral intervention. So occupational therapies, language, depending on what the problem is. So they don't have to go just by themselves, but they had to be implemented together.
Okay. Another question? Okay.
Thank you for the opportunities, Professor. Let me introduce myself. My name is Nisrina, and I actually want to continue a bit from Ms. Arina's question and your answer. So I'd like to start from my experience a little bit, not on experience, something that I experienced in life. So I have niece, And she has a very complex developmental disorder. And we've gone to many doctors, and they're not able to pinpoint a single cause or a single disease that she has. And so I also watch in a lot of YouTube, for example, that they're saying that not every developmental disorders are already characterized. And he also said that, for example, autism can be caused by several things. And so from a diagnostic standpoint, where do you start when you're faced with some disorder that maybe don't follow a conventional pattern of disorder, for example, and also when you're, for example, seeing some new symptoms, some new gait, some new trait, where do you start in the diagnostics?
Well, that's the challenges when you have a kid that has some problems that doesn't match with what we know, right? That could fall into the categories of rare disorders. And so that's what I meant, the odyssey of the family that bounce from one doctor to another to try to find an answer. And so I've been actually in a meeting a month ago in the Netherlands where now they are starting to put together all these databases. I might get, if you email to me, I might get you the link, where they basically are trying to collect all data from everybody who wants to participate. So like they ask me, do you have patients? I mean, I'm not a clinician, but I can tell my clinician, can you have the data and put it in a database where you say, I describe these kids has, this malformation, physical malformation and they use data also with imaging, the three-dimensional phases, and then the physical, behavioral, and so they try to match. Then you might say, oh, I have this case that comes from, I don't know, from Italy, and it's the same, look like this, and there are the images, and try to put the data all around the world where you can get, because if you have only one case, you're not going to figure out, but you start to have 10 or 15 cases that they are across the world, they look like that, and then maybe there is a way to find out what is maybe the gene, or what is the cause of the disease. It cannot be not even the gene in particular, but maybe a particular genetic variation. And, but it is a challenge when you have and they can't figure out. But I mean, if the sequencing doesn't show anything, if the arrays doesn't show anything, and then I guess that's the unfortunately situation. But you can do those things. So you can, I mean, the sequencing can tell you quite a bit, particularly if you also do the trios. So when you do the kid and then you do the parents, and particularly if there is another child, a sibling, and then you can study if there is something, a pattern that link that particular disorder to a particular variation.
Thank you so much for the answer, Professor.
Okay, maybe that's the last question for the session. Thank you very much, Prof. Flora, for sharing your expertise today and give valuable insight to all of us today. So on behalf of Biotechnology Study Program and the organizing committee, we deeply appreciate your time and generosity in this session. And we also thank Professor Tri Indah Winarni from Faculty of Medicine for giving us opportunity to collaborate through this event. Thank you very much for your active engagement and enthusiasm for today's visiting lecture. And may this session inspire us all to continue exploring molecular biotechnology for diagnostic and therapeutic advancement. And once again, please join me in warm round of applause for Professor Flora Tassone. And thank you very much, and that concludes our lecture and discussion session. Now I'd like to return the floor to our MC, Vina. Wassalamualaikum warahmatullahi wabarakatuh. Please stay. We need to take the photo.
Okay, thank Thank you for Professor Flora Tanase. And then before we proceed to closing session, we will have a souvenir handover from the head of Biotechnology Study Program, Dr. Sri Pujianto, to Professor Flora Tassone as the token of appreciation for today's visiting lecture. We kindly invite Dr. Sri Pujianto and Professor Tassone to come forward for the souvenir handover and photo session. Get in position, everyone, and the photo will start in 1, 2, 3. Maybe once more. Everyone get in place, and 1, 2, 3. Okay. Okay, everyone, for this photo session, we will do undip hands gesture. So, hands on our undip bermartabat di dadanya di sebelah kiri kanan. Undip bermartabat. 1, 2. Undip bermanfaat. 1, 2. Undip jaya. 1, 2. Dan yang terakhir ada Undip juara. 1, 2. Thank you, everyone. Ini ditutup langsung atau duduk dulu? Ini udah. Iya.
Cek untuk ujian UTS Biotekes A, jangan lupa yang B juga, jangan lupa untuk presensi di luar. Jangan lupa untuk presensi ujian ya. Sebagai tanda hadir ujian, untuk ujiannya nanti take home ya, akan diumumkan oleh dosen masing-masing. Jangan lupa dikerjakan ujiannya, itu ujian ya. Kenapa? Soalnya untuk kelas Pak Puji sudah ya, nanti coba dicek di depan. Untuk kelas saya nanti saya kirimkan melalui koordinator mata kuliah. Louis ya. Apa nanya apa? Yo, mumpung.